Have there been any adverse effects seen in any of the 3 trials you mentioned at the top of the call?
Yeah, a really good question. I mean, the adverse events, and I would urge you to, you know, uh, look up the publications. Maybe we can provide a link to some of the publications later on. But, the adverse events from so maybe I can go through the 3 trials. So from the UPenn trial, there have been no major adverse events, just events that are typically associated with cancer immunotherapy, which include things like, inflammation, cytokine release syndrome. So those are fairly manageable in that context. In the context of the CRISPR therapeutics trial, which is for sickle cell and beta thalassemia, the adverse events have led to, are probably caused by a delay in engraftment. So remember, we're wiping out the patient's, white blood cell compartment, the conditioning regimen. So then when you're infusing the stem cells, there's a period of time before the edited stem cells completely engraft and they repopulate the patient's bone marrow. So in that interim period between, infusion and engraftment, patients have suffered some adverse events related to the absence of white blood cells. So these could be like infections or, some other hemolytic syndromes. In the context of the transplant trial that I mentioned that happened in China that was published in the New England Journal of Medicine, no real major, safety issues. But again, it's really early days. What the agency, the agencies are most concerned about, and here I'm referring to the FDA and the EMA, among others, is the potential for long-term, what's called insertional mutagenesis, or like, you edit the hematopoietic stem cell, that could lead to an off-target effect that could lead to the development of cancer. So when you're editing stem cells, that's always a concern that people are worried about. We haven't observed it in any of the trials to date, but the data isn't long enough. We have to observe these patients for maybe another 5 to 10 years just to make sure that it's really safe. But so far, the answer to the question is that there have been adverse events. Most of them are related to the conditioning and the other, effects of related to getting the treatment, but there have been no significant health or adverse events related to the CRISPR gene edited product itself.
Great, thank you. Here's another one that I want to make sure we get to. So, as an alternative, instead of strengthening the human immune system to fight cancer, this person is asking, could there also be a strategy to eliminate the ability of cancer cell genes to neutralize the human immune system? That's a great question.
Yeah, it's a really good question. I think, it really depends on the cancer itself. So cancer is really a heterogeneous disease. And what we're appreciating about cancer is that, each cancer is unique to the patient. So although, a patient may have, let's say, breast cancer, the mutations driving that breast cancer are going to be fundamentally different across patients. And so the point being that, the inhibitory mechanisms that are used by the cancer could be extremely diverse. And so in order to really use CRISPR to genetically edit the tumor cell to eliminate that, it's going to be extremely customizable and extremely personalized, which, I don't think we're there yet today. But if we become more astute and just more operationally expert in using CRISPR, I think that could be a possibility. But really, it's going to be limited by just the heterogeneity and the diversity of cancer itself. It's not really one disease; it's many diseases, which really, I think, limits the potential application to the questioner's comment.